Everyone, welcome to this episode of Science Speaks, a series from Science Era that discusses a variety of topics about science and life in the world of science. Today we have with us Dr. Robert Carnahan, PhD. We'll discuss with him the topic of COVID-19 immunity and vaccine development. Dr. Carnahan is an associate director. He's the associate director of Vanderbilt Vaccine Center and an associate professor in pediatrics and radiology at Vanderbilt University Medical Center. He has a high level of experience with monoclonal antibodies and antibody engineering. He directed the Vanderbilt Antibody and Protein Resource for over 10 years. The facility grew to comprise over 70 Vanderbilt investigators and several academic and industry partners from across the US, conducting around 150 antibody projects per year. Both academic and industry projects focused heavily on therapeutic targets like cancer, viral pathogens, autoimmunity, and diabetes. Dr. Carnahan has brought this focus on innovation applied to generation and detailed characterization of antibodies to the Crow Lab and the Vanderbilt Vaccine Center. During this time, he has also held numerous regional and national leadership roles within the Association of Biomolecular Resource Facilities to develop advances in methodologies and technical standards, oversee collaborative studies, and implement training opportunities for lab directors and personnel. He is also a national leader in the design and implementation of lean management systems for biological laboratories. So welcome, Dr. Carnahan. Thank you so much for taking the time to chat with us. We really appreciate it.
Sure. Happy to be here.
Yeah, thank you. So before we dive into the topics of COVID-19 immunity and vaccine development, could you give us a quick overview of the work done by Vanderbilt Vaccine Center? I recall that in the past, the center has successfully developed monoclonal antibodies against a number of other viruses like Ebola, influenza, and Zika virus. So could you talk a little bit about it?
Yeah, so the Vanderbilt Vaccine Center does tend to focus more on monoclonal antibody development than it does vaccines, though developing antibodies is actually a really useful activity for even vaccine development, because by understanding the human immune response and what kind of antibodies are generated to pathogens, that gives us a deeper understanding of what a good response looks like and the kind of response we would want to elicit with a good vaccine. So we see antibodies and vaccines as having their own utility independent of one another, but they also have a strong synergy with one another, and understanding each side of the coin helps one to design the other, right? And so that's where we're at. We're at the interface of antibodies, understanding human antibody response, and how that's, again, useful for its own purposes, and then how that informs vaccine design. We do focus almost exclusively on viral pathogens, though we work on some other pathogens as well. But definitely we operate in the infectious disease space. We've made antibodies to probably at this point 20 or more viral pathogens and Ebola and others. And then some of these have already made it into some stage of clinical development. Our Marburg antibody was shown to be efficacious and is actually being stockpiled by the U.S. government as a medical countermeasure to Marburg. The Zika antibodies are currently in preclinical development of the manufacturing process for future clinical trials. Chikungunya antibody was used in a clinical trial a couple of years ago. And then influenza is kind of one of our hallmarks. We continue to operate in the influenza space, looking for broader and broader anti-influenza antibodies.
So that's thank you for that overview, Dr. Carnahan. Really appreciate it. It's very comprehensive and succinct at the same time. So I was hoping we can kick off our discussion by talking about the pathophysiology and immunology aspects of COVID-19 for a bit. As we are still learning more about the mechanism of how this virus gets people sick, scientists have been investigating what part of the pathophysiology or what part of the disease is caused by the viral infection itself versus what aspects were a result of a dysfunctional immune response, right? So on the one hand, it seems like you need to have a strong immune response to fight off the infection, and seems like groups with robust immune responses, like young adults, for example, have better outcomes. On the other hand, we have had recent reports of drugs that overall suppress immune responses, like corticosteroids, reducing COVID-19 mortality in patients who are very sick. And so we also had I think a recent study published last week, I think, that showed that the risk of death in patients with higher levels of biomarkers that characterize inflammation, like interleukin-6 or tumor necrosis factor alpha. So those with these elevated levels of these cytokines, their risk of death was more than double that of patients who did not have these higher levels of cytokines. So how do we reconcile that, or what does this tell us, and what impact does it have on treatment decisions?
Sure. Let me qualify anything I say by saying I'm a pretty focused, narrowly focused basic scientist. So I will speculate, but we would have to talk with clinicians and translational scientists to get a better feel for, pathophysiology and appropriate interventions. But I think what we're learning is that in most ways, this virus, unlike some of the spikes in like uncertainty and fear, it is not that unusual in most respects. It works pretty much like we would expect, like any other virus. You get infected, there's a certain course of viral disease, and then there's an antibody and an overall immune response, which generally clears it, and you're fine. And like with most coronaviruses, the symptoms are quite mild. The body is, and for most cases, people are perfectly capable of responding and clearing the virus. The differences, I think, that we see that are pretty narrow but important is, one, the transmissibility of this virus is pretty high, and so it's easy for it to transmit. We still don't completely understand the transmission, like what are the important factors. We understand some of them, but we don't understand all of them. And the other thing is, in a pretty defined subset of people, there is this secondary, it's almost like there's a secondary illness. There's the viral course, and there's an inflammation course, as you said. And what we don't totally understand is what the connecting point between those is. Like, what causes someone to transfer from a normal viral process into this sort of hyper-inflammatory process? And I think we're still trying to understand that. We know that it seems clear that it has to do not just with the virus, but the person. We see certain populations, like the elderly, people with diabetes and other underlying comorbidities, seem to be more prone to this, but it's still hard to understand the exact correlation. So, but if we separate those out, I would say, the antibodies and vaccines can help either way, because it does seem like it's the viral process that leads to the inflammation process. So if we can intervene early enough during the viral part of the cycle, antivirals will have an effect. We see this with remdesivir, right? The trials seem to say it's a limited benefit. It's not massive, but it does have a benefit. But the benefit seems to be, I would say the data there's still a lot of data to be generated, but it seems to be early, right? If you can get this, of course, I'm sure if we knew ahead of time, we could give someone remdesivir, that would be a little tricky to be able to maintain that. But, if you can intervene early after infection while the viral part of the cycle is still at play, then that can have a benefit. If it goes beyond that, then remdesivir doesn't have as much benefit, may potentially not any benefit at all if someone sort of made that transition from virus over to inflammation. So we know the viral process is the starting point, and suppressing that with vaccines through natural immunity, or antibodies through sort of engineered immunity, we'll call it, or small molecules like remdesivir, we can kind of probably stop or limit that transition to inflammation. Where things like dexamethasone and immunosuppressants come in is probably in that second phase, where now the inflammation and immune system are kind of got themselves stuck in a little bit of a negative loop where there's some bad stuff going on. The virus at that point is either gone or at least very low. We don't know. What we don't know is if you had a really effective antiviral, would it still have an effect? I would say remdesivir would not be a good example because it's kind of a general antiviral. A good monoclonal antibody might still have an effect because there might be low levels of virus at that point. But in general, we'd say it's not virus driven at that point. It's inflammation driven. So something that can just basically, it's like a fire. You want to have the firemen come in and just tamp that thing down so that the body can start to recover. And that's where I think, if you gave steroids early, like when someone's mounting an immune response, that probably wouldn't be beneficial. It's more in that secondary.
Backfire even.
Yeah, it might be bad. So I think that's clear. It's one thing for people to understand is, I won't say they're two different illnesses, but there's two distinct, to me, it looks like there's two distinct phases which need their own medical countermeasure. And those medical countermeasures will be different, possibly.
And I guess that, thank you. That's an excellent answer. A great explanation there. And I think one of the points you made about the analogy about the firemen coming in and putting out the fire. So that probably explains why the corticosteroid benefit that we have seen is also in very sick patients in much later stages, and to reduce mortality sort of as a last resort, right? So thank you, appreciate that. I think one thing I wanted to talk about was one of the concepts in immunology that have come up in recent discussions about the fight against COVID-19 is herd immunity. And so I was thinking, can you talk a little bit about why herd immunity is important against an infection and how it can be achieved?
Sure. I mean, yeah, herd immunity is an incredibly important public health concept. Herd immunity is where we basically get to a point where I would say defining herd immunity is specific to the pathogen. What level of immunity is needed to, quote, achieve herd immunity depends on how transmissible the pathogen is, how it transmits, and other factors related to the specific pathogen and sort of its life cycle. But the idea being that if you can reach a certain point of immunity across the population, I won't say guarantee, but you severely limit the ability of spread within the community, right? Because the transmissibility meets up where, if there's one person infected, there's enough immunity around them combined with the chance of transmission that you basically kind of eliminate it. If you have a really, really transmissible pathogen like measles, you have to have an incredibly high percentage of people immune for it not to transmit because it's so transmissible. Whereas other viruses that don't transmit so well or transmit through alternate sort of more complicated mechanisms, maybe you don't need as high a percentage. So yes, but I will say one thing people need to understand is the general way we achieve herd immunity is through vaccines, not through natural infection. We can do it. I mean, through the history of mankind, it's been through natural infection, right? That's the traditional, quote, traditional way to do it. And that can be achieved here. But if we look at COVID-19, which I would say the studies look pretty conclusive now that the mortality rate is somewhere between 0.5% and 1% of all people infected will die. We would have to say to achieve herd immunity, let's say herd immunity is 70%. Well, you would take 70% of the United States, whatever that number is, something over 200 million people, and then 1% of those would die as a sort of a progression towards herd immunity in the absence of a vaccine. So then we're talking about several million people. Now that's just bulb. I don't know if that's the real number, but that's the kind of reality we have to at least admit could be possible. So that's where, yeah, herd immunity in this case, we really want to achieve that through vaccines. We've seen places like Sweden and other countries sort of tinker with this idea of herd immunity. And what you see is that the rate of the virus is still so low that for us to openly achieve herd immunity is going to take a really long time. And so that's not something that's going to happen in the next few years through natural processes. So we hope that we can do it through a vaccine process, and then some medical countermeasures. Vaccines would have holes, right? Maybe they don't work in every population equally well. Maybe the immunosuppressed vaccines don't work well. Maybe in infants, maybe in the very elderly. So that's where we would have antibodies like the ones we and many other people have now developed, or small molecules like remdesivir, dexamethasone. We need all these. It's like, one thing everyone needs to understand is most viruses and pathogens, Zika, for example, or Ebola, we're to a point now where we just ran an Ebola clinical trial in the Congo last year after people working very, very hard for more than a decade or more on countermeasures. And we got to a point where we had a very sophisticated strategy that was able to be deployed and actually was quite successful. Here, we don't have 10 years. We don't even have 10 months. People are trying to learn everything about a new pathogen and develop countermeasures and make them and deploy them and understand them all at the same time. And that's definitely not how things work normally. It's not how things work best, but we're all trying to do the best we can in a really difficult situation.
I think that breakneck pace. I heard one of our Vanderbilt alums, Dr. Ted Ross, mention about it. He called it building a plane while you're trying to fly it.
Absolutely.
Thank you for that explanation on herd immunity, and I'm glad you talked about the human costs associated with the strategy where you sort of let the infection spread unchecked, and a lot of people getting infected instead of trying to achieve herd immunity via a vaccine, and in the meantime take measures to prevent spread as much as possible. So that's a great point. I want to go back for a second, if you don't mind, to talk about as we just mentioned previously, there is a wide range of variability and disease severity with COVID-19. It looks like, with some individuals being asymptomatic, and then a lot of them getting sick but mounting an immune response and recovering, versus the infection being more severe or even fatal for others. So, then there are, there are of course these recent reports talking about or confirming cases of reinfections or recurring infections. So how do these factors come into play, or how do they affect how we may design therapies or develop a vaccine against COVID-19?
Sure. So I will say, fortunately for us, this is not the worst case scenario. I know that people find it hard to but this is not at all the worst case scenario. We actually know a lot about SARS-CoV-2 because it is quite similar to SARS-CoV-1, and it's also similar to other coronaviruses. And so far, at every step, we kind of go through this. Again, people are seeing science play out in real time, which is not normal.
Right.
Usually we have multiple studies, we learn over time, it refines. Science is a great process for refining and getting better. But like all processes, in the midst of it, it's a little messy, right? Where people put on the table, and then we have to all look at those ideas and see if we can confirm them or not. So, but so far, I will say we have these moments where it's like, oh, maybe SARS-CoV-2 is different than all the coronaviruses. And then enough studies come out like, oh, no, actually, it's the same. And then another moment, it's not the same. Oh, it's the same. And so I would say what we'll probably find out is mostly it's the same. We know that other coronaviruses, even seasonal, we can induce immunity. It can wane over time. That's normal. It still lasts years, even when it's naturally infected. So we could predict that a vaccine will induce immunity. It looks like in all the animal models and all the other coronaviruses, that's going to be true. And with COVID-2, I strongly believe that'll be true. We could also suspect that maybe it'll wane over time. That wouldn't be unusual. I'm not worried about the current publications about reinfection. One thing that people need to mention that kind of doesn't get highlighted is that so far all the people, yes, they were technically reinfected. We could detect virus after it was cleared, and then virus reappeared in the system, and we were able to test it by very, very sensitive PCR methods. Keep that in mind. And two, so far, not any of those people have manifested any particular symptoms upon reinfection, which again would be normal. They have an immune memory, probably made up of both B cell and T cell memory. They get reinfected. Most of us clear it with the memory so quickly we couldn't even detect that they have been reinfected. Though that's probably quite common with all kinds of pathogens, right? We get it, we've had it, our body immediately responds, and it's gone before we can even detect it, let alone have symptoms. Here we have a few individuals which we were able to intervene, probably because we're so hyper aware and testing is so broad. It's actually like a consequence of our focus, not so much the immune system. So in places where they have really, really good surveillance systems like Hong Kong, they were able to detect that this person had virus present again, but they did not have symptoms. So that just means that their immune system has detected it. It had a memory response. Maybe it took a minute for the memory to sort of ramp back up, but it did, and I think that'll be the norm. It's not like we won't have some immunosuppressed people who can get reinfected and actually have symptoms on second exposure. I won't say that's impossible, but that won't be the norm. The norm probably almost certainly is going to be, yes, it's technically possible. It probably happens, and it won't be a big deal. So that means that our vaccine, even if it only lasts 3 or 4 years, I think we'll all take a 4-year vaccine at this point, right? We would all gladly say, okay, the vaccine's going to work for 4 years and then you need immunized again. I think we would all be like, okay, I'll sign up for that. So I don't think people need to worry too much about it.
Right. I mean, yeah, flu shots work seasonally, right? And we still, like a large chunk of us still are like, okay, it's not worth getting it and risking, especially in elderly population or somebody that's immunocompromised. So you still get it every year, even if the protection is only for a few months or to a year. So yeah, that's a great point. And I think it really helps people hear that the symptoms or the reinfection that we hear so much about is happens with other pathogens as well, and in this case too, like in the cases the symptoms were cleared or the virus was cleared pretty quickly. So that's definitely a good sign, as opposed to I guess we do hear a lot of reports about, oh no, like if is the reinfection happening? Is this going to be really bad? And like you said, science happening in real time, and you're thinking, oh, is this virus so unique? Is it so different? So that's thank you for providing that context. I know we touched a little bit upon the monoclonal antibodies and immune-based therapies. So speaking of those therapies, I know the team led by you and Dr. James Crow have recently discovered some monoclonal antibodies that are promising candidates for prevention and treatment of COVID-19, I believe. So I think you published the findings last month, right? And some of these antibodies were licensed by AstraZeneca. Where do you see such antibodies fit in the big picture of managing the COVID-19 pandemic? Can you talk a little bit about that?
Sure. Yeah, I mean, again, with everything playing out in real time, it's a little difficult to say. I would say in the midst of uncertainty, we should try to pursue all options. So there's no doubt that we should be pursuing small molecules, vaccines, antibodies, complementary therapies like corticosteroids. All those should be on the table. If we have what we really all hope for, even us, which is a efficacious vaccine, that would be one case. So one could ask, well, in the context of an efficacious vaccine, what role would antibodies play? Because vaccines would provide longer term immunity, probably at least years, if not lifetime. Antibodies won't do that. Antibodies are probably going to provide 6 to 12 months of protection. What role would antibodies play in that context? Well, antibodies would still have a role. We have to remember that the vaccine, even if it's efficacious, may not be equally efficacious in all populations. Maybe it doesn't work in pregnant females. Maybe it doesn't work in people over the age of 80, because you do need a functional immune system for the vaccine to work.
Yeah.
It basically, the vaccine is saying, this is what the virus looks like, and then your body says, oh, okay, I'm going to mount a response to that thing. If you can't do that, well, then that's a problem. Antibodies, on the other hand, what we've done there is sort of gone fishing in someone's immune system who successfully recovered, and we say, well, what antibody or antibodies allowed them to recover? And then we can just pluck that out of there, produce it in a factory, and give it. No need for, in general, much immune response on the part of the person. Now, of course, you need some complementary immunity around the antibody, but it's a much less of a burden on the receiver of the antibody. They don't necessarily have to have a highly functional immune system for the antibody to work. So in there, you'd say, oh, there are going to be holes possibly in the vaccine where an antibody or other therapies would be delivered as an alternative, or even in addition to a vaccine. It may not be either or. Well, this is still an emerging part of vaccinology, but there's also some ideas out there that a vaccine and an antibody together are actually mutually beneficial, that the antibody actually can stoke up the vaccine's response, but that's a whole separate thing. So there could be holes in the vaccine. Let's say we have a vaccine that is not as efficacious. Let's talk about another thing, post-exposure. So you have prophylaxis. That's where vaccines sort of are shine the brightest, preventing from getting illness. Now we have something called post-exposure prophylaxis. Now, let's say you've been in a room with 4 other people and one of them had COVID-19, and you talked with them face-to-face for more than 15 minutes without a mask because you thought that they were not, you was a family member. Now you don't know if you have the virus, but you've been exposed, right? You might already have it. At that point, vaccine may or may not be helpful, because the vaccine response takes 14 to 30 days to play itself out. If you even had a vaccine right then, it may not help. Antibodies shine really brightly there, because literally within a few hours, maybe even an hour of getting the antibody in your arm, you have circulating antibody protecting you from the virus. So even if there was an exposure, it would shut the viral process down pretty early. So that's a better spot for antibodies. Now we move to therapy. Now you're 2 days into your infection process. Could antibodies work there? Our initial data in animal models says yes, the antibodies still can be efficacious early in the viral infection process, where now we are sure vaccines are probably not going to be very helpful. Antibodies could be helpful. And we would have to see in humans, because one of the problems with our current models is none of them play out along the timelines of humans. They're all accelerated. All the animal models, they're too fast. Humans play out slower, so we actually don't know how many days into the infection process an antiviral antibody or antiviral drug would work. But we do know that their initial data with convalescent plasma and remdesivir suggests that antivirals do have a window once you're already having that infection pathology, that they can help. And then, like I said earlier, so that might be a sweet spot for antibodies, which I think will be more efficacious than small molecules, just because they're more specific. They have super high affinity. Right. They're also their half-life. You have to remember, a single dose of an antibody, you can get it once, and it's probably going to protect you for months, if not a year. Whereas something like a small molecule would have to be dosed daily, because their half-life is just so quick. But again, they both have their own roles. And then we go into the inflammation process. Probably antivirals may not have as big a role, but we don't know whether antibodies in a late stage might work. The convalescent plasma data is not great, but there is some suggestion that even in those later inflammatory stages, convalescent plasma has some impact. So that's to say that there's still some effect if you have good antibodies. Even when you're in this inflammatory state, the antibodies seem to be able to help. They can't make it go away, but they do seem like they improve things a little bit. But the data is still being generated there.
Yeah, I think that's a great overview. And so the molecules that have been licensed to AstraZeneca, are they in clinical trials at this point? The monoclonal antibodies?
Sure. They're in what's called phase 1 clinical trial, which is the initial safety study, and then looking at like what's called PK, pharmacokinetics, like what's the distribution, and so those early stages. But like everything, I mean, there's contingency plans to rush it, forward if it looks promising. And I don't have any of that data, but it's in phase 1 right now.
Gotcha. And I just remembered, you said going fishing for antibodies, and that sort of stretching that analogy a little bit seems like, directly giving monoclonal antibodies is giving a man a fish to eat right away, versus the vaccine is basically teaching him how to fish, but you may not have time if that person is starving. You need to give him food right away, and I just thought about that.
That's a great. I use that analogy too. It's a great analogy for
I know. So we talked a little bit about vaccines. So I know a number of vaccine candidates are currently in clinical trials, with some of them already in phase 3. The last part of the trials, right? And I was thinking, could you give a little bit of perspective about the top vaccine candidates that are in phase 3 now? Like I don't have to go into detail for all of them, but at least give a little bit of overview of what types of vaccine candidates are being tested. And are they, right now planning to have these as a single dose, where we only need to get the vaccine once, or do we have multiple shots, or even have booster doses at a later point? How are these planned? Could you talk a little bit about that?
Sure. Yeah, I can't give a comprehensive because there's so many. I mean, I think there are over 300 vaccines. Right. But I'll just talk about, this will be a U.S. sort of centric as well. So what we see here, though I think it's kind of mirrored elsewhere, is some of the lead molecules are what are called mRNA-based vaccines. So this is where, so if we think of the central dogma, DNA, RNA, protein, rather than giving them protein or some end product, which is a lot what traditional vaccines have been around, either giving a piece of the virus or an activated virus or a protein from the virus, we're moving upstream and giving them a coding, a message. The message is administered and it gets taken up into cells, and actually the cells in the body make the antigen, the part that stimulates the immune system, and they kick it out and it circulates. It's a really cool technology. Moderna and I believe Pfizer and BioNTech and CureVac and a bunch of companies have been hammering away at this technology for years. And we've worked with some of those, just for full disclosure, around antibodies and vaccines. And they're the most ahead out there because the technology lends itself to rapid development, rapid manufacturing. And those look like they'll be some of the first out. The initial data for those looks promising. It does look like, in all the cases I've seen at least, it will be multiple doses. So you get a single, and then maybe 15 to 30 days later, you get a booster to kind of stimulate the immune system. That would make sense because the mRNA, again, is dependent on the host cell to make the antigen. So rather than giving sort of a giant dose of antigen, you're kind of relying on the human immune system to sort of produce it, and maybe that doesn't generate as much antigen early on, so it takes a little bit of time, maybe needs boosted, but ultimately you arrive at a point where there is immunity. I think that's one group of vaccines. There's a kind of related ones where they're using adenovirus to kind of do a similar thing. You're basically incorporating the coding region of the antigen into an adenoviral vector that gets into cells, and now you're kind of doing the same thing. It's being made by the host cell via a viral vector that doesn't cause a long-term viral infection or anything. In fact, it's usually not species matched, so it's kind of self-limiting. It can only infect once. It can't infect another cell. But what you do there is you're kind of you're juicing up the like kind of goosing the system to make more antigen. And again, very limited data. It looks like that it does work a little faster. Maybe you only need one dose of that. Though it's, there's some that look like they didn't work well at all. There's one out of China that didn't look like it worked well at all, but like AstraZeneca has one of those, and the initial data looked quite promising that maybe a single immunization would be sufficient. those, I would say collectively, we all have to keep in mind that none of those have ever really been through a full FDA licensure. So, we think they'll work. There's lots of sort of R&D and preclinical work around those saying that they should work well, but we've never actually released those in a large scale.
So those are like really cool concepts and technologies, but have not been really established in a way like when we think of other protein-based vaccines or attenuated vaccines and so on.
Right. And those are shining because some of their key strengths is their flexibility. You can develop them quickly and rapidly and deploy them. Then we sort of move to the more traditional vaccine formats like protein. There's a company in the United States being supported called Novavax, which is developing spike protein, spike protein, and they're moving quite rapidly. But when you're making a protein, it just takes more time. There's more QC. I feel confident that will almost surely work. Probably with one immunization, but we don't have any, I think, data to know for sure that's true. At least I haven't seen any. But that'll be kind of lagging behind there by a couple months. And then not so much in the United States, but I know other countries, China being one and maybe some other ones, are looking at like inactivated versions of the virus as a possible, that's another traditional vaccine platform, which will probably work. But again, now we have to think, now we're growing up large amounts of virus, and then we're inactivating it. So that's the manufacturing there is more complicated, more dangerous, requires a lot more control. So we're going to have multiple it's going to be a weird time where, usually you have sort of one vaccine or two vaccines to choose from. You know a lot about them. Here we're going to have a lot of vaccines that work from very, very different technological perspectives, have, different delivery strategies, different regimens for like how often you get it. So, it's going to be a bureaucratic, we have to really have our government agencies really, really engaged in thinking about this. It's a really complicated problem that we have the ability to solve, but it's just we have to be out ahead of it because, it's going to be a little complicated.
Absolutely. So, as you said, there are a lot of these candidates right now, and some of them may even be ready to administer around the same time. So is that a good thing for a pandemic that's of this scale, and especially given the variability? I would imagine in that case having more options would be helpful. Is that accurate? Or
yeah, I mean, I guess it's kind of like, to use a gambling analogy. We want to put our bets down on as many things as we can, and some of them may not work well, but at least we would have some that work. Maybe even some work not well, some work medium, some work awesome. So at least in that first burst of activity of immunization, we've gotten some people well protected and some people intermediately protected. That's way better strategy than say, I'm going to put it all on red number 5, and then it doesn't turn out to be that one, and nobody's.
Yeah,
I do think we're doing the right thing in sort of betting on different technologies, betting on different delivery strategies. We will have to acknowledge the fact that unlike in the normal world where we would know which ones work best, here we'll figure it out when it's used, and that's not an ideal situation. I do think that the safety profile of all of them look pretty good, maybe even excellent in most cases. So I don't think we're necessarily risking safety, though I do think, I will say I'm a strong advocate that the phase 3 clinical trial needs to run its full process. That's how we know in a large number of people, rare events didn't happen. The phase 1 trial can be in as few as 50 people, right? That's not a very strong demonstration. That's an initial safety demonstration, very initial. But if you have an event that's happening at 1 in 1,000 or 1 in 10,000, have to run the phase 3 trial to see that kind of event. And that's the more typical kind of adverse events we see with drugs and vaccines and every kind of thing is they're not 1 in 100, they're 1 in 1,000 or 1 in 10,000. And so you got to let those trials run so that we can not only see it, but we can understand it. We need to see it more than once to say, was it an individual, something about that individual that, so it's not something for us to generally be worried about. It was really kind of a unique situation of an individual, or more worrisome, we need to see it multiple times. Was it a population? What if in 70, we have to do 30 or 40,000 people to get enough people over the age of 70 to say, oh, you know what? In people over 70, there's this adverse event that happens 10% of the time. You got to have enough numbers to power that, and you got to let the clinical trial process play itself out. We're already expediting crazily. We don't.
Right. Yeah, the pace is just, I think, breathtaking to me. Like usually we say vaccines could take 3 to 4 years, maybe even more, right? And so a lot of these candidates, when we say, it'll be ready in a year or even less, that's that just seems just so wild, especially given some of those, right? Like when we, as you said, some of those molecules, if you see adverse effects, you need to have that strength of several thousands of patients to detect that 1 in 1,000 event or 1 in 2,000 event, and sometimes it happens much later after, like in post-marketing studies, right? So I think, that's a great point, Dr. Carnahan. So you mentioned, it is known, so there are some populations that are at more risk than others, as you said. So we may need a specific, to go for a specific separate vaccine for them, or a higher dose, like we have with flu. So are there any specific studies or analyses being conducted or planned in higher risk groups, like older populations or those with comorbidities, for example, that you're aware of?
Well, again, we're kind of in this non-traditional space, and I will say I'm for sure not a clinical trial expert. I just, I don't know much more than the general person just from reading a little bit. What you see is a lot of the people who are the companies moving forward vaccines they're really trying to be diligent. Everyone has to understand, no one wants an adverse event less than them. They really don't want one, not just for financial reasons, but like the entire company is based on these. They're all working hard. These are really serious scientists who really want to contribute. They don't want an adverse event. So what you see them doing, I specifically heard this one with Moderna, but I know other companies are doing, is they're trying to push into some of these populations where there might be issues, or at least where we've seen worse outcomes, like African Americans, the elderly. They're trying to specifically enroll people in those populations to detect these events. Is there something there? Is there certain kinds of comorbidities where the vaccine doesn't work very well? So that's just an efficacy question, but there could be an adverse event question too. Is there certain populations where there are adverse events? As you kind of said earlier, a lot of this stuff, we phase 3, but then we have post-marketing where we continue to monitor these adverse events, which a lot of times are going to happen later. So we're not going to have that when we start to probably roll out the vaccine.
Right.
We do. So the fastest vaccine of licensure in the history of the world, I believe, is 4 years. That's because they did the phase 3 clinical trial, then they waited a while, they let things happen. The average is decades. Vaccines generally take decades for full licensure and release, because companies want to be sensitive to this, like, well, what happens to all these subpopulations initially and then over time? If we want to resolve the situation, we'll all have to agree that we're going to take on a little risk as a society. We don't know exactly. I think it's quite low risks, but some risk that we don't get to do that. But, we do have to balance how much risk are we willing to take.
Right.
To gain a month is probably not worth it to take much risk. To gain years, these post-marketing studies will probably have to take some risk on. But, right. So that's where we're sort of like trying to balance it all out.
I heard there is a little bit of a push from some of these vaccine makers, the pharmaceutical companies themselves, to make sure that the clinical trials are, truly running their course and do their due diligence, and they're not, pushed by regulatory agencies, or they're not submitting these to regulatory agencies until they believe their candidate is truly ready.
Their reputation, it's not just they want to help, they're trying to contribute. They're also, the reputation of their company goes beyond just the vaccine. If the vaccine works, if the vaccine doesn't work and has numerous adverse events, that's a tremendous problem for them as an entire company across all the things they work on. So.
Right, absolutely. So, and you said, so far it looks like from what data we have seen of these candidates, the safety profile looks overall good, which is a good sign, right? But the vaccine might be differently efficacious in people, especially given the variability of how the disease affects everyone. So there is a concern that deploying a weekly effective vaccine could actually make the COVID-19 pandemic worse, especially if authorities or vaccinated individuals wrongly assume that it makes them completely immune, right? Leading to sort of a complacency or a reduction in other COVID-19 prevention or control measures. How valid is this concern?
Well, we will see. That's where we have to have to me, and again, a non-expert, we got to have the full phase 3 data in front of us. What kind of immunity did it generate? How reliable was that? Was it 90% of the time there was a robust immune response, in which case we can all feel pretty good? Was it 52% of the time, in which case exactly, now we start getting the problem like, oh, it's a 50-50 shot of someone's. Then we would have to say, do we still deploy the vaccine? Yes. But now it becomes really important that we have an assay to determine the efficacy of the vaccine on an individual basis. So there you'd say it just becomes a strategic question. I don't think it's a game change. It's not a stopper. It just is Then you have to say, okay, Ram, you're going to come in. I'm going to give you your initial. You're going to come back in a month later. I'm going to give you your boost. Then you're going to come back in a month later. I'm going to take your blood and I'm going to run an assay and say, yes, you look like you're good, or no, you don't. And then in the context of that, again, another strategic thing would be we would start to pull back on the prevention measures in accordance with what we're seeing at that assay. Once we say, 75% of the United States has gotten their dual immunizations and they've gotten their screen and it looks like they're mostly protected. Now we would have, again, we ramped up stage 1, 2, 3, 4. That's how we did it in Nashville. I don't know how you did it where you are. So then we should also be clear that it should go down 4, 3, 2, 1, or however, it should go backwards. It shouldn't just be like, hey, we have a vaccine. Magic is here. Let's go. Everybody.
Right. Yeah, I think that's a great point, especially given the COVID-19 vaccine is on track to become the fastest developed vaccine in history, right? And so it is. I definitely agree that it's really important to have clinical trials run their course, and we make sure, really evaluate how much the different candidates work. And instead of, like you say, oh, this is a magic bullet, we are good. It almost feels like, this is still taking a lot of time. And for a lot of people, like you said earlier, it's science happening or unraveling in front of their eyes in real time, right? And so suddenly, they're like, oh, 18 months is too long, or even a year is too long. So do you have an idea of, how close are we to a safe and effective COVID-19 vaccine, or what is a realistic timeline you think for making a safe and effective vaccine available to the public?
Right. Well, I would say how we should ask that the clinical trial process needs to play out, and that dictates the timeline. Again, you've heard people say over and over, I think, who are more knowledgeable about this than me, that the clinical trials that people look like they're launching and the rate at which they're accruing patients looks like they will have accrued a sufficient number of people for their phase 3 in later this year, something like November or December. Then there'll be a time period at which they analyze the data and we all sort of pick it apart. It needs to be reviewed by someone outside that organization. That's how science works.
Right.
Science is a self-correcting process. So it's not like just because the data's there, we all say it's okay. The FDA needs to see it, but other people, independent parties, need to review it just to make sure that everyone agrees. And that's all predicated on the fact that we don't see, again, a totally normal thing to happen in a clinical trial. Let's give you an example, is that something bad happens to an individual patient. That person has a severe response. That person maybe dies. That is a normal event, because you have to remember, we're doing 30,000 people. In the context of a clinical trial, it's very likely someone's going to die amongst 30,000 people. Now, the question is, did their death or adverse response tell us something about the vaccine? One completely normal answer would be no, it actually had nothing to do with the vaccine. That was something that would have happened, or it's not very related to the vaccine. It's not generalizable to the population. But what happens is you have to pause the phase 3 and investigate that. That's the safe and normal thing to do. Come back with a conclusion and say it was or wasn't related to the intervention, and then move forward or not. So all of these talk of November, December is predicated on the fact that we don't have any of that. But that's a totally normal event in a phase. That's totally normal. It's expected. You have a lot of people involved with all kinds of different health issues that have nothing to do with the vaccine. So anyway, even if we get through that unscathed, there are no adverse events that we have to slow down. Then we have to talk about, even the governments across the world have been prepaying for manufacturing, so the stuff's already being made, even though we don't know if it works, which is a good strategy. I'm not against that. But even if we had a warehouse full of an efficacious vaccine on December 1st, how long does it take to get that down to a level where non-frontline people, non-enhanced people who don't need it first get it at a Walgreens. They walk into a Walgreens and or CVS or whatever, or their local healthcare clinic and get it. That's a distribution issue, and it has to be done correctly. Especially, just think about what you said. If it's 2 doses, then you have to keep track of the timing between the doses, and we have to just know that itself is going to take months to play out. I think we're thinking, a realistic expectation to me would be in March or April of next year, a somewhat large percentage of the population will be receiving the vaccine around that time. That would be if everything goes perfectly. People receiving the vaccine on a large scale in 2020, I don't see that lining up with the timelines, except maybe in some 1% perfect world, which we have to be careful of that world, because it might be a world.
It could backfire too, right? Yeah, we want to do our due diligence, like you said, and sort of going off of that, what are some of the potential issues that can rise if we rush a vaccine to market, like less effective one, or even worse, like something that has potential harmful effects? Would this, lead to decrease in overall confidence in vaccination, or like not just for COVID, but for other universal vaccination as well?
Yeah, you hit it exactly on the head. You have the sort of what we'll call the proximal issues, I'll say, the short-term ones. The short-term ones are, we have people who have serious side effects, and we have to deal with that, and that's not good for any of us, right? In the context of a pandemic, now we also have layered on another layer of complexity of people having adverse events, and then does that tie to particular comorbidities or populations or whatever? So they have this short-term problem, which would be, again, a big burden in the context of what we're dealing with. Equal or bigger does that overall, we know that adverse events affect all of us, how we think about things, right? We still are, or even non-adverse events, just bad publicity or bad studies, right? The MMR vaccine, I've pointed out to people, we still have a ongoing issue with vaccine resistance that tries to studies which were retracted and were not done in a correct way, or not interpreted in a correct way. But that's had a tremendous impact on vaccination, not just with MMR. That's the weird thing. People have to remember is that, like, we shouldn't think about it that way, but people think of vaccines as a thing. They shouldn't. Like, vaccines aren't really a one thing. That's like saying cancer drugs is a thing. Well, like cancer drugs, like there's all kinds of different cancers, there's all kinds of different drugs. But people tend to think of vaccines as like this monolithic thing, like they all work the same way. They don't. They work by completely different mechanisms. They work at different levels of efficacy and all kinds of things. So, but the problem is, since they have that name on them, vaccine, a failure of a vaccine in one space can affect, people's willingness to entertain a vaccine in another space. So, there's the real harm, is that we rush out a vaccine. It turns out to be low efficacy. Flu, we see this. We're always fighting this battle that like people still get the flu when they have a flu vaccine, and they're like, vaccines don't work. I got the flu vaccine. I still got the flu. It's like, well, let's be cautious to say that like flu vaccines are kind of guesswork, because flu's constantly changing. We're guessing. Right. When you have the right one, it works well. It's just we have no way to necessarily know with the certainty that you got the right one. But that doesn't mean that you weren't protected all the times you didn't get it when you did get it.
Right, that's true.
You cannot remember this because you don't remember it because you didn't get the flu. So, but you do remember the 1 year you did get the flu.
You did, yeah, that's true. We do have that sort of selective memory, and unfortunately, a lot of public health interventions too work very similarly, right? Like we look back and salt, like iodine in salt, or like fluoride in water, like any of those stuff. We don't think about it, but it's working well. And the one time any of this stuff doesn't work, we immediately go to be like, wait a minute, like that did not work for me. And so yeah, it is unfortunate that like you said, it can have that long lasting repercussions. And so I'm really hoping like the vaccine trials run their course, and we get a good candidate, or maybe multiple good candidates that can be mass produced and given the people in a safe and effective manner. And in the meantime, we don't push for, like you said, herd immunity is a very important concept. It needs to be achieved for the population, but typically it's done via vaccination. So we cannot, like you said, 200 million people being infected, and even 0.5% mortality of that is 1 million, right? So it's that's such a heavy cost, and so we probably need to be more aware of that, and really take preventive measures as necessary, and let science take its course. So we have covered quite a few topics today, and I know we're coming up on the hour. So this has been a really great discussion. Really appreciate you taking the time today to share your insights with us, and providing us all these information. So on behalf of Science-Era, myself, and our viewers, thank you very much, Dr. Carnahan. We really appreciate it.
Sure, that was great. Thank you.
Thank you.